What's up, baby bio penguins? So we are doing our FRQ Friday. I know, so exciting. So what I've decided to do is for the next 4 weeks, we're going to go live, and we're going to go over the FRQ questions from the 2021 exam. So today we're going to do FRQ 1, and then next week we'll do FRQ 2. Following week we'll do FRQ 3 and 4, and the following week we'll do 5 and 6. I was going to go live on YouTube, but it just seemed a little bit easier, and I just know how to work Instagram Live to do these. So yes, I'm in my bathroom because my son is sleeping, and I'm trying to kind of help y'all while also, being a good mom. So without further ado, we are going to flip the camera, and we're going to talk about FRQ from the 2021 exam number 1. So we have this polycystic disease. That's an inherited disease. It causes water loss from the body, and it affects the cell division in these kidneys. So what you're going to see on the exams, they're going to give you some type of disease or some type of experiment. There's something that they're going to bring to you, and they're then going to explain about it. So you're going to not have to know about these diseases. You're not going to have to know about these different inherited things, but they're going to give you what you need to know. So because the water movement across the cell membranes is related to ion movement, scientists investigated the role of the sodium-potassium ATPase, also known as the sodium-potassium pump, in this disease. So we have this albumin, which is a steroid hormone, and it binds to the sodium-potassium ATPase in plasma membranes. Individuals with the PKD have a genetic mutation that results in increased binding of the albumin to the sodium-potassium ATPase. The scientists treated normal kidney cells with the PKD cells with the increasing concentrations of this albumin hormone, and then measure the number of cells. And then we'll see that in figure 1. And then they'll do the activity of the sodium-potassium ATPase, kind of the activity of that enzyme, in figure 2. And then we'll see the hypothesized signal transduction pathway in figure 3. They're going to look at this MEK and this ERK, and we're going to see how those all interact together. So here you can kind of see figure 1. So figure 1 is looking at the albumin. So we can kind of see as the concentration increases, what's going to happen to the number of cells. And so you can kind of see that it increases to an extent, and then it does kind of drop off and level off at some point. Okay. You do also look at these error bars. Notice which one of those are overlapping. So we see at this really low concentration that those error bars overlap. But as we kind of get into these larger concentrations, those error bars stop overlapping. And that error bar tells us whether our data is statistically significant. So if they do overlap, that means the data is not statistically significant. The differences we see here is due to chance and chance alone. Versus over here, what we see, those differences are due to the experiment because of the fact that, PKD actually does have an effect on the number of cells and the way that it's binding with that albumin hormone. Okay, so here we see figure 2. We can see how the concentration levels, the same concentration we already looked at with figure 1, how those concentration levels are going to affect the activity of the ATPase, okay? And then over here we see in figure 3, we're looking at that signal transduction pathway of where the you have the receptor of this sodium-potassium ATPase is going to bind to that hormone, causing some type of conformational shape change that then leads us into this transduction pathway, right? The MEK gets activated to become phosphorylated, so we have the PMEK, and then that activated PMEK will then activate the ERK to PERK, right? So it also gets phosphorylated. So you're just seeing a phosphorylation cascade in here. And then that PERK is going to move into our nuclear envelope. So it kind of goes into the nucleus, and then it acts kind of as a transcription factor to turn on the transcription of a certain gene, right? So that's all the information that was given to you to begin with, right? So they're going to give you tons of information. And what you need to do is kind of figure out what of that information do you really need and what kind of do you need to bring forward. And for some reason, my computer keeps advancing. So if I keep clicking the slide back, just go with it for a second, okay? So our first question we have, the A question, is always going to be a question that's asking your biology knowledge. So what biology stuff do you know that applies to this, okay? So our first question is going to be, describe the characteristics of the plasma membrane that prevent simple diffusion of sodium potassium across the membrane. So you have to think to yourself, okay, what do I know about the membrane? I know the membrane is made of phospholipids. Okay, cool. Okay, what is a phospholipid? A phospholipid is made up of a phosphate group, and it's got fatty acids. Okay, cool. Okay, so how is it oriented? Well, I'm going to see that I have my phosphate heads facing the interior of the cell and the exterior of the cell, because they're polar. Versus I'm going to have those fatty acid tails kind of facing the interior of the membrane, right? They're going to be on the inside of the membrane facing one another because we have this bilayer. So I know that my plasma membrane is ambipathic, right? Where I've got the phosphate heads are polar, facing the exterior and the interior of the cell. And I know that my fatty acid tails are nonpolar, facing the interior. So the outside of the membrane will be polar and the inside of the membrane will be nonpolar. Okay. And so that's the first point that we see here. That's the first point you could have gotten. You could have accepted any of these answers. So the interior is hydrophobic or nonpolar. The phospholipid tails are going to be hydrophobic. The exterior of the plasma membrane is hydrophilic, or you could have talked about that the heads were hydrophilic. Okay. So that's all. Yeah, you'll be able to watch this later. I'm going to save it and I'll also post it onto YouTube later. So now we kind of have to think about, okay, the second question is explain how ATP is required for this. Okay. So, well, sodium and potassium are charged. So because sodium and potassium are charged, they're unable to move across the membrane without some type of assistance, right? Now, because of the fact that I need ATP, that tells me that it must be going against its concentration gradient. So right here we see that explain why ATP is required for the activity. Well, sodium potassium ATPase pumps ions against their concentration gradient, and pumping them against their concentration gradient means that it requires this metabolic energy. It requires the ATP to provide that energy that's needed. Okay. And so that's all part A, right? So there's 2 points given for part A. 1, could you describe the characteristics of the plasma membrane? 2, can you explain why ATP is even needed? Which is very biology, right? If you know your bio content, you can easily get that A point, right? So the second question, the B part, is going to talk about the specific experiment and talk about the different components that make up that experiment, okay? So let's look at what they're asking us to do for this one. So they want you to identify a dependent variable in the experiment presented in figure 1. So if you look at figure 1, we say, okay, what did we change? We as the scientists changed that albumin, right? We changed the concentration of that hormone. And how does changing the concentration of that hormone, what does that do? Well, that's going to change the number of cells. So my dependent variable is going to just be the number of cells. Yeah, you got a point for looking at this axis and saying, hey, look, that is what my dependent variable is, okay? So remember, there are points that are easy points, right? There's points where if you don't know anything, you can look at a diagram and try, right? Even if you got it wrong, you should at least try. You should write something that's in that diagram and say, okay, maybe it's the concentration of the albumin. Maybe it's the number of cells, right? But remember, we have 3 different diagrams. Make sure you're using the correct figure when you're looking at this. So we were specifically looking at figure 1. So pick one of the axes and try to guess, right? Don't ever leave something that is given to you in a picture blank. It's okay if you're wrong, but it's worse if you just don't answer, right? If you don't answer the question, then you're setting yourself up for not getting the point, right? Okay, so the second question we have to say is justify the use of normal human cells as a control in this experiment. So why do I need there to be human cells? Like, why do I even care about using these human cells? Well, the whole point of the experiment was to see whether this disease is going to affect the concentration of, the number of cells and the activity of the ATPase, right? So if I don't have the normal cells, I have nothing to compare it to, right? So it allows the scientists to determine the effect of that disease on cells, and it allows them to compare the responses that we see with those cells, right? So you always have to think about what is the point of a control. Now, be very careful when you're writing your responses that you don't just say, well, it's to compare to the treatment group. Be specific about your experiment. What is happening in the prompts? What happened in the experiment they brought up that you can bring in? We're observing the effect of the albumin on the number of cells. Like, we are observing how that difference is between the disease cell and the non-disease cell, okay? Make sure you show them that you know what you're talking about, okay? So here is the scoring guideline, right? So it says it allows the scientists to determine the effect of PKD on the cell's responses to different concentrations of albumin. Now keep in mind that anywhere where you see like a parentheses, that means that's an optional thing you could say. You don't have to say the stuff that's in parentheses. Okay. Another thing you could have said is allow scientists to compare the responses of the PKD cells to the normal cells. So by just saying it allows you to compare, you've said the right information, but you're not specific enough. So you have to make sure that you are specific when you're writing your responses, okay? And our third thing is justify the use of a range of the Obermann concentration in the experiment represented in 1. So when we look at the graph, we see that we go from 0, we go to 1, we have 10 to the first, second, third, fourth, fifth, sixth, seventh, right? So why do I have this concentration? Why do I keep increasing the concentration, right? So they're determining whether different concentrations affect it. And they actually, as scientists, may not know exactly what level is going to see a difference, right? So if they've never done this experiment before, they've never tested this hormone on the cells and seen the effect, they're getting a large variety of concentrations, that logarithmic scale, so that they can see what do the different concentrations do, because they personally don't actually know the answer. That's what happens in science. You don't already know the answers. You just have to do experiments to find the answers. So the scientists need to determine whether different concentrations have different effects on the cell numbers. And the scientists did not know at which concentration the Obermann would be in effect. Okay. So that's B, right? So C is going to require you to analyze. You've got to somehow interpret some data, right? So use your graphs. The answer is going to be in the graph. Now, when they say to analyze, you have to make sure that you're not just restating something you see in the graph. Okay, so you have to bring a little bit of biology into it. Okay, so the question is, based on the data shown in figure 2, describe the relationship between the concentration of Obermann and the the sodium potassium ATPase activity between the normal cells and the PKD cells. So if I look here, okay, and I see, okay, well, as that concentration increases, what do I see happening to NHKD? Well, I see that it's decreasing. As the concentration increases, the activity decreases. And the same thing that I see with the PKD is as the concentration of the Obermann increases, as I get this number larger, then I see that my graph is going to decrease. I have a negative slope, okay? So I do a little circle there for you. So increasing concentration of the Obermann is going to result in decreasing ATPase activity in both of our cells. You could also say that it's an inverse relationship, or you can say that there's a negative correlation between the concentration of the Obermann and the ATPase activity. Okay. So just make sure that you are directly connecting the Obermann to the percent of the activity. I've had students before where they connect the wrong thing. Like, as the activity increases, the concentration decreases. Well, yes, that is a true statement, but it's not because of the activity that the Obermann is changing. So you have to be very careful the way that you word these questions. So there's a question in the chat. It says, do we have to answer the FRQ parts in order? Like, if we're short on time, can we just answer the graph-related questions? Of course. So when you're writing your response, make sure that you label your parts. So part A, part B, part C. But if you want to answer C first, just write C and write out your answer for C. And then write A and write your answer for A. It's perfectly fine to write them out of order. You do not have to leave like space to fill them in later. We as the readers are perfectly capable to read through your entire response and get those points for you. Okay, even if it's out of order. Also, if you don't label it, you will still get credit. It's just easier for us as the readers. We grade thousands of these responses. So if it's kind of you're parting out A and you go ahead and write it, it's just a little easier, and it kind of breaks up the first response a little bit more and helps to keep us a little bit more aware and kind of focused. Like, yes, we'll read your response as many times as it takes to get the points, but it just kind of helps us a little bit better if it's broken up like we ask it to, like with the ABCD. So we answered that. Okay, so the second question we have is, almost lost the phone there. The scientists determined that the sodium potassium ATPase activity of the PKD cells treated with the 1 picomolar Obermann is 150 units of ATP hydrolyzed per second. Calculate the expected sodium potassium ATPase, oops, my computer's running low, ATPase activity in PKD cells treated with the 10 to the 6 Obermann. So if we look at our graph and we say, okay, well, where's 10 to the 6? So 10 to the 6 shows me that I have a 30% activity. So they told me that 1 is 150 units. So I take 150 times 30%, which gives me 45. Okay. So the answer is 45, or you could have said any number that's between 40 and 50. Don't give them a range of 40 to 50. Do give them a specific number. That range is provided just in case you didn't read it exactly like the author of the question did. It provides just a little bit of variance so that they can ensure that you can get your points, because you clearly know your biology. You clearly know how to read and analyze. Now, notice that was a calculation, okay? I believe there's probably going to be at least one calculate on every single exam, okay? At least one of your FRQs will have a calculate. So those of you that are a little bit scared of your calculator, y'all, you can do this. Take deep breaths. Just do your best. If you don't know, just try, okay? So, wait, why do you times by 150? So we multiply by 150 because if I look at the question, it told me that the activity treated with 1 picomolar is 150 units of ATP hydrolyzed per second. So since I'm looking at over here where it was 100%, oh, I'm sorry, I was zoomed in the wrong spot. So since I'm looking over here at 100%, right, you would say, okay, well, if 100% is 150, then 30% is only going to be 45 because it was 30% when I was at 10 to the 6. I hope that made sense for the person who asked the question here. Wait, did someone ask a question in the chat? There's too many questions from my story, so I actually can't find where it is. Are all papers coming internationally read or in the U.S. or, okay. So the person asked about the international papers. All international papers are also read by us. So we'll go to Kansas City and we will grade all of your responses. There's different people who will grade the different parts. All right, so part D, because my computer is running low on battery and I need to make sure that I at least get the FRQ done for you. So part D is saying, okay, well, in a third experiment, there was another experiment. The scientists added an inhibitor of phosphorylated MEK, which we call PMEK, to the PKD cells exposed to 10 to the 4th picomolar albumin. Based on the figure, predict the change in the relative ratio of ERK to PERK of the albumin-treated PKD cells with the inhibitor compared with the albumin-treated PKD cells without the inhibitor. Y'all, these questions. Oh my God, there was so many letters in that thing. So if we're looking at the graph, right, and we say, okay, come on, flip. Okay, so if we're saying, okay, well, if there's an inhibitor of the PMEK, right, then this step right here won't take place. If that step right there does not take place, you no longer have the enzyme that speeds up this reaction, right? So the ERK to the PERK is not happening, right? It's not increased, it's not speeding it up, all that fun stuff, right? So you would expect there to be a higher concentration of your ERK and less of your PERK, because this process of converting ERK to PERK is not taking place, or is taking place a lot slower of a process, okay? So you would expect there to be a higher ratio of ERK to PERK in the cells with the inhibitor because of the fact that they're unable to convert it over, which you also are supposed to justify. So there's our justification. The amount of PERK will not increase as it does in cells without the inhibitor. The amount of ERK will not decrease as it does with the inhibitor. The cell continues to make that ERK without making the PERK. And then PERK is not being dephosphorylated to make ERK. This question. Oh my. So that was what we could have said for our justification. So then the very last point we have on this is using the data in figure 1 and the signal transduction pathway represented in figure 3. Explain how the concentration of the cyclin proteins may increase the PKD cells treated with the 10 to the 4th picomolar albumin. Okay. So when we're looking at this, right, we're trying to figure out, okay, well, what does cyclin do? Cyclin's job is part of the cell cycle, right? And so if you have a higher amount of your cyclin, right, because we know we have cyclin-dependent kinase, right? If you have a higher concentration of cyclin, you're going to go through the cell cycle more frequently. So if we look at our graph, we see, okay, here's 10 to the 4th. I have a very high concentration of cells. So there's a high amount of division that's taking place here, right? So I see this increase in number of cells because of this albumin, right? So I look at the pathway. The albumin has this whole signal transduction, right? Which then causes the transcription of target genes, right? Those target genes are probably cyclin, okay? So what we're seeing here is that the cell gets to this maximum of 10 to the fourth, and the signal transduction pathway stimulates the transcription of genes, specifically those that are needed for cell division, which that increase of cell division genes is going to include the cyclins, which regulate the cell cycle to increase that activity. So you're bringing together the experiment as well as you're bringing in your biology knowledge. And yes, the question does seem to get harder as you go down. If you know your biology, A is pretty easy. B is bringing information from the diagram, bringing in a little bit of experimental design. Sorry, that's C. C is bringing in the diagram, and then D is really where they hit you, right? They get really complicated on you, okay? So if you're trying to renumber your questions, you could, if you don't know A, just jump to B, get B and C, come back to A and try that one out, and then just give your best on D. Okay, so that is all of I have on the FRQ Friday for today. So, we will come back together next Friday. We'll do number 2, okay, which will be our graphing question. So if you want to get prepared and kind of say, okay, let me do this question on my own before Ms. Jones goes live, go for it, right? Go ahead and answer that question, see what you think the answer is, and then when I go live, you can get your paper out and see whether you did well on it, whether you understood the information, or if you had trouble with it, you can maybe get some of those questions answered. So do we have any questions before I log off of the live? I haven't heard my son yet, so I'm assuming he's still sleeping, which is good. So what's your advice on avoiding confusion when there's a lot of acronyms in a question, like the one we just had? It's very hard to not get confused. So for me, when I was doing this question, I kind of had to like follow it through with my finger. Like, okay, so there's the IRK and the PERK. Okay. And then double check your writing. So as a student, you might write the wrong word. As a reader, I read what you write. So if you wrote the wrong word, even if you know the right word, I can't give you credit. So make sure that after you've done a whole bunch of those acronyms, go back and reread it and make sure that it says what you think it says. Make sure that it says what you want it to say. Your free responses, they say 10 minutes for a short FRQ and they say 25 minutes for a long FRQ. I would try to sit at about 20 minutes for your long FRQs. That gives you 5 minutes to go back and reread over it. Make sure there's nothing that you missed. Make sure you didn't accidentally say the wrong word. It just gives you that breathing time to go over it. So my students all year have only gotten 20 minutes on their long FRQs. I kind of have cut them off at the knees, and they actually can write FRQs in a lot shorter than that. So is there any questions in the chat before I log off? Going once, going twice. I'm going to say sold to the, no one's writing me. Remember, AP Bio Penguins are dressed for success, and I will see y'all next week. Bye, y'all.